Hello everyone, my name is Altinalmazis-Kondylis and this is a pre-recorded presentation on the effect of tree mixtures and water availability on below-ground complementarity of fine roots of birch and pine planted on a sandy potsoil. This work was recently accepted for publication at Plant and Soil Journal. Mixed species plantations are considered one of the main options for adapting to and reducing risks of climate change. This is mainly because tree species mixtures have been found to be more resistant to adverse climatic conditions and to biotic hazards than monospecific stands. In addition to their higher capacity of mixed forests to stabilize key ecosystem functions, such as primary productivity, an increasing number of studies found that tree diversity increased plant biomass through overyielding. Yet, even though scientists and foresters are interested in above-ground overyielding, as higher timber production is desirable, little is known about the below-ground adaptations involved in overyielding, and if they vary along the soil profile. Previous studies have given contrasted results on below-ground overyielding. Some studies have reported positive outcome of tree diversity, and it was hypothesized that whenever below-ground overyielding occurred, that was due to better soil volume filling due to different root traits of the involved tree species or due to vertical root segregation. However, mixtures of tree species that fulfill these requirements have also given neutral or even negative results in other studies. For example, in Schmidt and Kata 2002 and Bolton and Villanueva 2006 studies, there was positive and negative outcome of diversity respectively. However, vertical root segregation was observed for both studies. Such discrepancies in the outcome of tree diversity may be due to the variability in the environmental context, such as precipitation, soil fertility, or soil structure, which can all play an important role in explaining the root distribution across contrasting ecosystems, especially since benefits of species mixing may be more pronounced on nutrient-poor and dry sites than on rich and wet sites. With that in mind, our main objective was to evaluate the interactive effects of species mixture and water supply on fine root distribution and morphology, and assess whether trees overyield below ground when growing in mixture. To achieve this objective, we worked in the Orphy tree species diversity experiment in southwestern France, which was established to investigate biodiversity and ecosystem functioning under the influence of a changing climate. In 2008, a total of 25,600 trees of 5 native species were planted within a 12-hectare area in a randomized block design, giving a total of 32 species combinations. Since 2015, half of the blocks receive irrigation of the sprinkler system every night during the summer, and the other half experience the dry summer of southwestern France. An experimental setup which allows us to study the interaction between tree diversity and water supply. From all the possible species combinations that Orphy has to offer, we chose to work with birch and pine monocultures and their mixture, as birch and pine are considered to have a priori contrasting vertical root distributions and fine root morphological traits, with the possibility to stratify further under mixture. Each plot of our experiment contains 10 rows of 10 trees, planted at 2 meters apart, resulting in 100 trees per plot, with a plot area of 400 square meters. The soil is a typical potsoil with coarse texture, acidic conditions, and is very poor in phosphorus. More importantly, this soil is characterized by a discontinuous cemented spotty horizon, a hardpan at 50 centimeters deep, and also a fluctuating water table that goes shallow, ranging from 40 to 80 centimeters during the rainy winter time, but drops between 150 and 180 centimeters during the summer months. For our experiment, we harvested litter and soil cores at 90 centimeters deep from 4 sampling points within its plot. Each soil core was then further divided into 5 soil layers of different depths to better capture the vertical root distributions of roots. From each of these layers, root samples was then extracted, scanned to assess fine root morphology, then dried and weighed. In order to assess the direction of diversity effects, we chose to work with the relative yield metric, which is calculated for each component species and is the division of observed yield of species A in mixture divided by the mean yield of species A in monoculture. Values that are below 0.5 equal to 0.5 or above 0.5 indicates negative, neutral, or positive mixture effects. This relative yield can further be calculated for the whole community by summing the two relative yields of the two species, resulting in a relative yield total. The reference value for the relative yield total, similar to relative yield, was 1. In order to assess vertical distributions, we chose to work with the beta parameter from Gale and Gringal 1987, which was determined by nonlinear regression for each entire soil column. The beta parameter gives values that range between 0 and 1. Values that are close to 0 indicate a shallower fine root distribution, while values closer to 1 indicate a deeper fine root distribution. To that extent, we tested three hypotheses. First, we hypothesized that mixtures of trees with contrasting root rates should result in vertical root segregation and higher total relative yield. This is because pine and birch present contrasting root rates, which might in fact lead to a release of the competitive pressure and the more efficient soil volume occupation. For instance, pine is reported to grow rather deeply and has thicker roots, while birch has more extensive superficial root systems that are finer and well ramified. Secondly, we tested the hypothesis that vertical root segregation and below-ground overyielding should be larger in plots which are not irrigated and thus potentially experience summer droughts. This is because when tree species grow in mixed stands and under favorable growth conditions, for example with increased water availability, interspecific competition should be likely weaker, while under harsher conditions, for example under decreased water availability, interspecific competition would be stronger. In the case that the competition is strong, the less competitive species may respond with a shift towards less occupied niches. For our third hypothesis, we hypothesized that shifts in soil space occupation would follow a shallow-rooted versus deep-rooted pattern. We expected that birch would grow better in topsoil layers, while pine would grow better in deeper soil layers. This is because the fine root of birch are much finer in diameter than pine, and should be able to explore and exploit favorable growth conditions, such as the ones that exist in topsoil layers, more rapidly. Alternatively, because pine roots are thicker and have a more conservative growth strategy, they might be able to grow better into less favorable soil layers, such as dense soil layers. If we take an overview of the vertical root profile, we see that for both tree species, fine root biomass decreased with increasing soil depth. However, we observed that birch was more plastic than pine in response to irrigation and mixture. Birch actually responded with a significantly higher fine root mass under irrigation and a lower fine root mass under mixture, with the clear differences observed for the shallower soil layers. On the contrary, pine did not respond significantly to irrigation or mixture. Regarding fine root morphology, specific root length and specific root area had the highest values at the forest floor and significantly decreased with soil depth for both species. As expected, birch had higher values than pine, but they were only marginally affected by irrigation and mixture, and there was no clear morphological adaptations based on the entire fine root class and for each soil layer. The three species in our study were previously reported to have contrasted fine root distributions and fine root morphologies, with the potential to stratify further under mixture. Contrary to our first hypothesis, the vertical root distributions of pine and birch were relatively similar along the soil profile, and there was no below-ground overyielding. Actually, both species had very shallow root systems in both pure and mixed stands. 80 to 87% of roots were in the upper 30 centimeters of the soil profile. The similar and shallow root systems may be due to the cemented layer observed around 50 centimeters deep that may have forced both tree species to use what is remaining of the soil profile in shallower layers. Moreover, for short periods of time, the water table could reach minus 30 to minus 40 centimeters below the soil, while in the summer it does not necessarily always descend below 120 centimeters. Such fluctuations of the water table can inhibit fine root proliferation into deeper zones, creating anoxic conditions while at the same time stimulating the proliferation of fine roots in the layers above the water table and near the soil surface. Contrary to our second hypothesis, vertical root segregation was not larger under ambient water supply. In particular, we find that in the control treatment, designated by the red line, birch and pine had almost identical vertical root distributions, while under irrigation, designated by the blue line, birch produced shallower roots than pine, resulting into an apparent vertical root segregation, which exists only under irrigation. Interestingly, we also found a marginally interactive effect of between mixture and irrigation, as under ambient water supply, both mixed tree species distributed roots deeper than their respective irrigated stands, while in the pure stands, the opposite occurred and the trees distributed their fine roots to shallower soil layers in response to ambient water supply. This result indicates that vertical root adaptations to water availability change within the specific competition. It might be that monocultures adapt less than mixtures due to negative feedback from species-specific pests and pathogens, or because mixing species mediates environmental stress. We know from previous studies the importance that interspecific competition has on vertical root distributions, as the inferior competitor might be forced to shift their roots to unoccupied soil space, causing vertical root segregation. However, in this case, we reported higher flexibility of the birch root system to water availability, particularly when irrigated during the growing season and when enduring interspecific competition. However, we should be cautious in our interpretation. Although root location is an honest indicator of water and nutrient uptake, it has been shown that the spatial distribution of the roots does not always correlate to the spatial distribution of uptake. So general tendencies of roots adapting towards deeper distributions or shallow distributions does not automatically imply that the two trees are actually competing hard for water and nutrients. Contrary to our second hypothesis, the relative yield total of the sum profile did not significantly change under different water supply, and there was no significant overyielding under neither control nor irrigated conditions at the community level. When looking at the individual tree species, we found that under ambient water supply, birch and pine tended to produce on average the same amount of total fine root biomass in mixed stands relative to their respective pure stands. However, when irrigated, birch produced 46% less fine root biomass in mixed stands, and pine produced 54% more fine root biomass. The general decline in birch fine root biomass in mixture may be due to the competitive superiority of the pine above ground. Pine had a larger basal area than birch, which implies larger needs for water. Hence, it would also take up water faster, grow faster, and outcompete the relatively slower growing birch trees, leaving less available water in the soil. In contrast to our third hypothesis, shifts in soil space occupation did not follow a shallow rooted versus deep-rooted pattern. When investigating per soil layer, we found that under ambient water supply, the relative yield for birch and pine was not significantly affected. These results suggest there is a rather symmetrical interspecific competition of birch and pine plantations along the soil profile when water availability is lower. On the contrary, under irrigation, we found that the interspecific competition was asymmetrical along the soil profile and favorable for pine. Birch roots were significantly underrepresented in most of the soil layers, and pine was significantly overrepresented for the layer of 0 to 5 centimeters. The general idea that interspecific competition is alleviated through fine root adaptations towards shallower or deeper layers has been described in previous studies, but this is not supported by our study. Our results highlight the importance of looking into different soil depths to better understand the net outcome of competition in mixtures along different environmental conditions. However, we must remain cautious in our interpretation as we don't know whether competitive ability is static or changes dynamically over seasons and years. It could be that interspecific competition could eventually lead to vertical niche partitioning when tree stands reach maturity. To conclude, our study revealed that 10 years after planting and after 3 years of summer irrigation, mixing silver birch and Martin pine did not affect vertical root distribution. It did not result in significant below-ground over-yielding considering the sum of the soil profile and their ambient water supply. This is probably due to the relatively high groundwater table and the presence of a harpine at 50 centimeters deep, which did not allow roots to explore deeper. Furthermore, the commonly used shallow versus deep paradigm was not present in these aggrading young forests, and evaluating the root profile in terms of shallower or deeper fine root distributions gave undecisive and unclear results. Instead, effects were specific to soil layers and depending on the environmental context, which could indicate that effects of interspecific competition may need more time to be visible along the whole soil profile.